Thank you for joining us for our latest presentation this afternoon. The presentation that we're about to have right now is focused on spot-on multiplexing. It's going to be mainly focusing on some of the future things that are going to be happening here at AutoGenomics and different tests, different instrumentation, different extraction methods, etc., that are coming in the near future. And that being said, we have several distinguished presenters today. First will be Fareed Qureshi. He's the President and CEO of AutoGenomics. He will be speaking first on multiplexed automation to meet the evolving lab needs. Following Fareed will be Dr. Jacques Corbeil, who is from the Canada Research. He's a Canada Research Chair from Laval University in Quebec. He will be presenting on two different presentations, but the first one will be the automated microarray serotyping of pneumonia, and the second presentation will be automated influenza typing as well. The third presentation will be from Bo Fessenden. He is the CEO of NMTC, and he'll be talking about an extraction method, so simplified extraction for automated molecular testing. So I ask that you hold your questions till the end, until the three speakers have spoken. At the end, we'll open it up for a Q&A session, and at that time, we'll have the panel speakers available to answer any questions that you may have. So that being said, I'm going to go ahead and pass this over to Fareed to begin our presentations.
I'm supposed to talk about automation, and that's really one of the wildest fields, and I have to condense that talk in 15 minutes, so it'll be very interesting. But automation by itself is an endless topic, really. And I think as you walk around in this conference, you will see almost every company talking about automation. But when you're talking about automation, the way we have approached, or AutoGenomics has approached automation, is really developed because there's no one type of automation which can fit every lab's need. And so what we are doing is really develop a series of platforms, and you'll see three of them on the floor right now, which addresses the different need of the lab and to make them really more efficient through their workflow and many other environment in the lab itself. We do realize that the labs are running like factories, and they need to be without any interruption and without even a thought going on what the next thing is happening. So that the future instruments, as they are coming out, addresses that type of environment, is the lab high throughput environment. We did not steal this from the airport TSA. We actually developed a technology which focuses on target and signal amplification. Okay, and we'll talk about that and how we use this technology in automation. We definitely have mastered the art of microarray processing. Very few microarrays have really made it into a commercial environment. And even though microarray by far are the most adapted in giving you multiple answers, and yet the manufacturing and the quality control of microarrays are the most tedious, and this is where we have excelled in AutoGenomics on that. We've really introduced a new form of the use of microarray. We have figured out, or actually invented, a method in which we can now tag a patient sample with a unique sequence and then use it on a microarray, allowing multiple patients to be run on a microarray. We'll talk about that. And then on the other hand, where there is more content required, where you really have to look at hundreds of genes all on one patient, we can use multiple microarray all on the same system and get a similar result on that. Now, when you're looking at automation, you're really looking at many things as how you can reduce your hands-on time, streamline the process, increase workflow, and reduce cost. And the tools which we have used is really how to use the electronics, the mechanical, the software. But what we have introduced here is how we have used the genetics in automation. We actually, by tagging a unique sequence to every patient, we are using the genetic information in order to streamline a process on that, which nobody else has achieved so far. So in order to automate the platforms, first of all, we have developed a biofilm chip, which is All of you are very familiar with the 35 millimeter film, where it conditioned with 21 different layers of emulsion in order to create an image. Well, we do the similar thing. We don't have 21 layers. We have actually 4 layers, and we can actually apply a biochemical sensor like a DNA or an antibody or any protein, and we can create an image of a sequence which is there or not there. Okay? We have completely automated the process in processing the entire microarray from sample to result. And in order to do that, we use a very unique method in which the reagents are actually monitored and administered. In order to do all of that, we had to develop a scheduling method which was not used or not known in the industry. Okay? We then packaged all of this from the operator interface to the sample management reagent, all the consumable, the detection system, the result analysis, all into one instrument. And I think you will, as you walk around, you will see that. Now, there are several complexity which normally in immunoassay or some of the other fields don't present, as you will see in the area of molecular diagnostic. In the area of molecular diagnostic, you are looking at many things in a sample. You're looking at sequence variances. You're looking at each SNP or single nucleotide polymorphism. You're looking at insertion, deletion, and repeats in the sequence. You're looking at expression to see how a certain gene is expressed, and then even protein identification and quantification. On top of that, the next level of complication comes on the type of samples. We use, if you look at, we have 42 different applications which we market to different part of the labs. And they use blood, tissue, different types of tissue, frozen blocks, urine. And so I can go on and the type of complexity of how you take the cell out of the sample present quite a challenge in doing molecular testing. Now, in order to take all of these into account and address how you can get not just 5 or 10 or 15 tests per day, but to achieve or develop a system where the results which you are getting is like maybe 1,000 or 2,000 tests per day. There are tests right now, especially dealing with infectious disease such as chlamydia, gonorrhea, trichmosis, or any kind of screening panel. The requirements, they are molecular, and the requirements are not 20 or 30 tests per day, but they are much, much more. They are 500 to a few thousand tests per day. So in order to do that, the first thing AutoGenomics had to address was a detection system which can really look at maybe few thousand scans or spots at one time. Okay, our microarray, we are able to put as much as 1,024 spots, and we had the task of really trying to scan 100 microarrays at one time, and those are the tasks. Those are the type of detection systems you had to come up with in order to achieve the throughput which the labs are now demanding in molecular. Okay, we had to develop a method in which we can use the microarray which has never been used before. By tagging a patient's sample with a genetic sequence and then assigning a zip code, we are able to use multiple patients on one microarray. We'll talk about that a little more detail. The next thing was the complexity of molecular automation is so high that an average scheduling system which just scheduled one task after the other was not going to be able to do the level of automation which we need. And so we had to develop a software scheduler which uses a queuing theory in order to align the task based upon the weight or the importance of that task. Okay? These three things all have to happen together in order to achieve an extremely high throughput molecular platform. Okay?